Hi dear students, today we're going to talk about polymerase chain reaction or PCR. As we all know, this particular topic is quite familiar to all of you because in different sessions we have come across the use of PCR. So I'm just going into the details of it in a faster mode. As we all know, this technique finds application in lots of purposes, either in medical and biological research labs. And you can also find PCR is being used in various diagnostic techniques, let it be in the food microbiology or in the medical microbiology. And also it finds applications in the forensic field where you need to multiply or prepare a large copies of a particular DNA. And the use of it is still extended to various other areas where you have to make or you need to identify a substance from a small segment of DNA, or sometimes from a small bone, or sometimes from a small hair, and all that. So wherever be the process of PCR is being used, the basic principle is just the same. That's because we make multiple copies of a small segment of DNA from the sample which we have obtained, and we check whether a particular gene of interest is there. Sometimes if you are dealing with screening of colonies, or sometimes you might be or to identify for the taxonomic identification of microbes, you might use PCR technique, or sometimes for the identification of any substance, whether it is a plant or whether it is an animal, you can use PCR to identify if COVID-19 is there. You can to check if you have COVID-19 by a PCR technique. That is also the PCR which we use. The same principle has been followed in all these techniques irrespective of the same principle. Now coming to PCR, the photocopying machine of DNA, it was developed in 1983 by Cardi Pcr. And the vast majority of PCR methods, they may normally use thermal cycling. That is, alternatively, we will heat and cool the PCR sample to a defined series of temperature steps. And the reaction, normally it requires DNA containing fragment, which has to be amplified. It could be genomic DNA or cDNA, whatever it is. Then you need two oligonucleotide primers which are specific to the DNA sequence of your desired fragment. You should have an enzyme called the purified DNA polymerase. Usually we use stacked DNA polymerase normally. And you should have DNTPs, which are the building blocks of DNA, and also a buffer solution which contains Mg2 plus and K plus cations could also be present. So this is a buffer mixture which is being needed for the reaction of PCR. Normally, the process of thermal cycling is being involved, and you follow three main steps. One is the process of denaturation, followed by a primer annealing step, and then finally a extension step. The denaturation is usually carried out at approximately 94 degrees centigrade, where the double-stranded DNA will get separated into single-stranded DNA, and approximately at 60 degrees Celsius, the primers, depending upon what purpose, you have different primers. So the primer will get bound to the single-stranded DNA. So this process is called the primer annealing step, which approximately is being carried out at 60 degrees centigrade, which will vary depending upon the primers. And finally, you have a extension step where multiple copies of your gene of interest or your fragment of DNA which you have to be amplified or multiplied, that one's extension will be carried out at 73 degrees centigrade. And after the process of extension, this cycle has been repeated again for about 25 to 35 cycles. So the process of denaturation, annealing and extension is being carried out 25 to 35 times, making multiple copies of your gene of interest. Now, repeated thermal cycling initiates new rounds of DNA replication, and that would cause the products of the previous round as a template, thus exponentially amplifying the target DNA fragment. So if you go to look into the process of PCR, you can see that a double-stranded DNA, it has is being present, and in the double-stranded DNA, you have a DNA fragment that has to be amplified. And while the process of denaturation is being carried out, the whole strand of DNA, regardless of which region and all that, the whole DNA gets denatured to be single-stranded DNAs. That is, the hydrogen bonds between them will get separated. And then annealing of the primers will be carried out. That is, I told you, at approximately 60 degrees centigrade, these are the primers. The primers will come and bind. So the primers will be at specific regions where the gene of interest or the gene those segments which has to be amplified. From that region, the head as well as the tail region, the complementary to that, we will synthesize primers and you will add primers. And once you add primers, you will also have DNA polymerase in the mixture. So DNA polymerase comes and binds to it, and then the process of DNA synthesis will be carried out, or the process of extension will be carried out. Now with the first segment, you can see the extension of the DNA fragment has been carried out at a longer phase. And in the second phase, what happens? Again, the process of denaturation will be carried out. So you get 4 different strands over here. And then the primers go and bind again, and the process of DNA synthesis will be carried out further. Then again, the process of denaturation will be carried out. You get single-stranded DNA. The primers anneal and DNA synthesis will be carried out. Like this, the process of denaturation, annealing and DNA synthesis will be further carried out again and again. And you can see that multiple copies of your gene of interest will be synthesized finally as a result of the polymerase reaction. Regarding the enzymes which have been used for the PCR, the most frequently used enzyme is Taq DNA polymerase, or the Taq polymerase simply speaking, which was being obtained from Thermus aquaticus. And we know that because we have to expose the DNA as at high temperatures, the enzymes which are being used during that particular temperature should be able to withstand the temperatures of about 94 degrees centigrade. And so, Taq DNA polymerase is something which can withstand high temperatures, but at the same time, it helps in the process of DNA replication also. But Taq DNA polymerase lacks a proofreading activity of about 5'-exonuclease activity, and it also has a higher error rate. That is 1 in 9,000 nucleotides are incorporated, 1 error appears. So there are alternatives for Taq DNA polymerase, like we have Pfu Polymerase, which has been obtained from Pyrococcus furiosus, and it has an optimum growth temperature of about 100 degrees centigrade, and so the protein which has been obtained from it, that is a Pfu polymerase, is something which can be used for PCR. And the error rate of Pfu polymerase has been found to be very less compared to Taq polymerase, and Pfu polymerase has a proofreading activity and has the lowest error rate of any known thermostable polymerases. So Pfu polymerase is ideally suited for PCR replications where high fidelity amplification of the DNA is being required. Now, this is the old 3 temperature thermal cycling Pcr. Now, of course, that was the old one, but right now there are different versions. You can just Google search, you will get different versions of the Pcr. This is also an old version of it, but you can come across different models of the same Pcr. But the basic principle is just the same. So you have a template DNA which is having, you will have an append of tube into which you will take your template DNA, your sequence specific primers, the DNA polymerase, the DNTPs as well as a PCR buffer. And you will set in the Pcr machine the different steps needed. That is initial denaturation, 94 to 96, 2 to 3 minutes depending upon your DNA sample. Denaturation further will be carried out for about 0.5 to 2 minutes for a longer denaturation. Then annealing will be carried out approximately at 60 degrees centigrade, then 72 degrees centigrade for the extrusion. So you have a first initial denaturation where the entire template DNA becomes single stranded. Usually, in the case of some DNA polymerases like PFU and all, they will require a hot start for activation. That's a technical portion. I'm not going to the details of that right now. So the first cycle 1, the step 1 which we have here is the initial denaturation. Then you have the cycle 2 which consists of 3 steps, and this process will be carried out further for 24 to 30 times. Then you have a terminal extension where incomplete products will be just finished. And this is a reaction, the example of a reaction mixture which you are preparing. So usually you will prepare either 25 microliter reactions or 50 microliter reactions depending upon how much sample you have to amplify and what is the purpose of your PCR. So these are about the primer dilutions. That is, when you get the primers, this is only at the technical point of view which I have included in my slide, so that anyone who's working on PCR and who wants to start with primer dilution, they can get used to it. Now coming to the variations of the PCR. There are different types of PCR. One is a Cardi Pcr, where you take a small fragment or a small speck of the DNA and not DNA, the colony, and directly you can use it for the PCR amplification and screening. So the colony samples are obtained with a sterile pipette tip or a toothpick and transferred onto the PCR mix, and without the cell disruption and all that, the PCR that is being carried out within the append of tube itself. You need not isolate the DNA beforehand. You will just take a small speck of the colony, and at the temperature of about 95 degrees centigrade, which is already used for PCR, the cell will rupture and the DNA will be released and the normal procedure will be carried out. So this is mainly used for the rapid screening of transformed colonies, especially in recombinant DNA technology, to check the presence of insert, its orientation as well as the size. And for screening desired recombinant clones from DNA libraries, thereby reducing the time and an effort required for screening large number of colonies. So the benefit of the Cardi Pcr is that you need not isolate DNA from every colony which you get on a recombinant plate after the process of transformation. This is what the procedure is. Pick a colony from the DNA library onto a microcentrifuge tube or a microtitobell. Add the PCR lysis reagent, vortex and heat at 5 minutes for 95 to 100 degrees centigrade, and run the PCR using an aliquot of the lysed cells and analyze the result by gel electrophoresis. Then another one that we have is a nested PCR. It is a modified form of PCR designated to reduce the contamination in product occurs mainly due to the non-specific amplification. So what you do is you will use outer primers as well as you will use inner primers. So the first PCR reaction will be carried out using the outer primers, and that amplified product will serve as a template for the next PCR, and inner primers can be used. Now this is a pictorial representation of that. You will use first outer primers and you will amplify. This is the first whole DNA, and from that you will amplify and you will get your desired gene, and then again you will use inner primers to check whether, and you will do a re-amplification. So you are checking whether the specific gene of interest is there in order to avoid contaminations as well as to be specific, like in this particular region, specifically this region is there, you're checking that. So that is about the nested PCR. Then you also have a multiplex Pcr where more than one target sequences can be amplified by using multiple primer pairs in a single reaction mixture. And since multiple genes are in the target in a single reaction, it is called a multiplex Pcr. And various information comes out from a single test run, otherwise it would require several reactions. And so what does it mean? You're using here multiple primers. So you look at this particular reaction. If I want to identify 4 different strains of a particular organism. Now, for example, a mycobacterium, okay, which are having different strains or something like that. I need to find out like that. I will prepare a primer which is specific for strain A, strain B, strain C, and strain D. And then I will put all these primers together along with the DNA sample and do a PCR amplification. And if this particular fragment, that is a fragment of this particular length, is been formed, I will say that it is of strain A. You must have an idea. The colors does not indicate that there are 4 different colors will be formed on the agarose gel. It only indicates that what happens, they will be of different lengths. Okay. So if one particular length of a fragment has been formed, you will say now this is green color, not green color, which has been found to be very much longer. That will be of strain C. Then if it is blue, size corresponding to the blue one, it is represents to strain B. Like that, you can identify which strain it is. So multiplex Pcr is normally used for pathogen identification, mutation analysis, gene deletion analysis, as well as in forensic studies. Now the next one is a reverse transcription Pcr called the RT-PCR. You take an RNA strand, and the RNA strand is reverse transcribed into its complementary DNA using the reverse transcriptase, and subsequently the cDNA is amplified using the PCR. Now in most of the viral infections where they have a genetic material of RNA, if you want to know their presence, what do? You take their RNA strand, you'll take the RNA and you will convert it into cDNA, and then you will add what? The primers, and check whether that particular pathogen is being present. Now, various enzymes are being used. Now, for example, you have enzymes which are being obtained, that is reverse transcriptase enzymes are being used over here. And these reverse transcriptase enzymes can be used from either, they have been isolated from avian myeloblastosis virus, Cardi Pcr virus, etc. And you can see that when you are synthesizing from RNA, you have synthesized the cDNA. And certain other primers such as oligo-DT primers or sequence-specific primers can be used further to amplify the cDNA. And sometimes thermostable DNA polymerases having reverse transcriptase activity, but which requires manganese as a cofactor, can also be used as instead of the reverse transcriptase enzyme. So the basic Pcr protocol is just the same, but only thing is that it requires an additional step in which the RNA has to be converted into cDNA. So RT-PCR, as we have told, you can use it for different purposes, either for the detection of viruses, RNA viruses, or you can also use it for genetic disorders to detect and also in obtaining eukaryotic exon sequences from mature mRNAs. So reverse transcriptase PCR, it mainly involves the conversion of RNA to what? cDNA, and then the cDNA is been amplified either by using non-specific primers like poly-AT primers, okay, yeah, poly-DT primers, DT primers over here, and or you can have specific primers also. This is the schematic representation of here. You are using the first strand synthesis, you have the mRNA. From the mRNA, you are synthesizing the cDNA, and then poly-DT, the DT primer is being used here. So in mRNA always you already have a poly A tail. So the primer will be poly DT primer here. So specific to this A, the poly T will go and bind and synthesis will be carried out. Or if this is one way where when you use the oligo DT primer, or another case if you're using a sequence specific primer, you can use the next one. That is, you can use a sequence specific primer and then synthesis will be carried out for the first strand synthesis, first strand cDNA synthesis. Then the procedure is just the same as the other one. The only thing is that you have RNA conversion to cDNA, and then either you can use a oligo DT primer or a sequence specific primer. We'll continue with the RAPD. That's also a modification of that, where the segments of DNA are amplified at random and no knowledge of the DNA sequence of the targeted gene is required over here. And usually RAPD markers are decamers or made up of 10 nucleotide lengths. And a single nucleotide primer will bind to many different loci. And so normally it is mainly used to find out, that is, whether different varieties are being present. Like if you have different varieties of a plant or of like that, they will have a RAPD pattern and you can identify them. The same procedure of RNA, Pcr amplification has been carried out, but the primers which you use here are being found to be random. So it is mainly used widely for the genetic diversity or polymorphism study, the genetic structure of populations, or for the detection of soma-clonal variations. Now, I guess you came across different types of Pcr reactions, and this is, now you have G1, G2, G3 and G4, genotype 1. So it will have a pattern like this. Genotype 2 means it will have a different type of pattern. So the basic protocol of Pcr has been followed over here, but the primers which are being found to be random over here, and they can bind at different things. So depending upon the pattern which you obtain, you can say that whether the different varieties which you have are of the same genotype or not. And whatever be the type of reaction that you carried out, Pcr optimization is always needed, because sometimes extraneous DNA can happen to come over here, as well as the amplification of DNA might not be present sometimes if too much of DNA is being present. So that process of Pcr amplification becomes quite essential, and primer designing techniques are also important to improve your Pcr reaction. And I hope you got a overline of the Pcr techniques in a brief way. So thank you one and all.